Hi everyone. In this lecture, we'll begin with transcription in eukaryotes. In the previous lectures, we have seen the transcription process in detail in bacteria, that is prokaryotes. Transcription in eukaryotes is much more complicated as compared to bacteria. The reason is that in bacteria, there is only one transcription enzyme, that is RNA polymerase, one type of RNA polymerase. In eukaryotes, there are three classes of RNA polymerases that carries out the transcription of various genes. Also, eukaryotes are complicated in context to transcription as compared to bacteria in the way the transcripts are processed in eukaryotes. Before we see these three types of RNA polymerases in eukaryotes, let's see the types of RNA molecules in eukaryotes. There are basically four main types of RNAs in eukaryotes. First, messenger RNA or mRNA that comes from the protein coding genes that can be translated into a polypeptide. Second, ribosomal RNA, which is part of the ribosomes. Third, transfer RNA that brings amino acids during the translation process of protein synthesis. And the first three types that we discussed now, mRNA, rRNA, and tRNA, are there in bacteria also. And the fourth main type of RNA in eukaryotes is snRNA, called small nuclear RNA, which we will discuss or talk about in one of the later lectures. So, three types of RNA polymerases doing the transcription of the genes that give rise to these four types of RNAs. RNA polymerase 1 synthesizes three types of ribosomal RNAs. 28S ribosomal RNA, 18S ribosomal RNA, and 5.8S ribosomal RNA. So out of the four ribosomal RNAs in eukaryotes, three of them, 28S, 18S, and 5.8S, are synthesized by RNA polymerase 1. Then we move on to RNA polymerase 3, which carries out the synthesis of 5S ribosomal RNA, which is the fourth ribosomal RNA, all the transfer RNAs that brings amino acids during the translation process, and some of the snRNA, which are not synthesized by RNA polymerase 2. And then we come to RNA polymerase 2, the enzyme that carries out the transcription of the protein coding genes, the genes that code for proteins, and synthesizes messenger RNAs. So RNA polymerase 2 synthesizes messenger RNAs and small nuclear RNAs. So three RNA polymerases, four types of RNA molecules, with three ribosomal RNAs synthesized by Pol1, the fourth ribosomal RNA synthesized by Pol3, tRNA synthesized by Pol3, and some snRNAs again by Pol3, snRNAs by Pol2, and transcription of the protein coding genes, resulting in the synthesis of mRNAs by RNA polymerase 2. We will see now in detail the transcription of the protein coding genes in eukaryotes by RNA polymerase 2. So eukaryotic genes also have a promoter. So what is a promoter sequence? We have discussed this in depth in the previous lectures, that promoter sequence is a sequence upstream of the coding sequence in a gene, where the RNA polymerase comes and orients itself to begin the transcription at the correct site. So this is the promoter sequence, and promoters are there in eukaryotic genes as well. And eukaryotic genes, which are transcribed by RNA polymerase 2, have a specific promoter sequence where the RNA polymerases will orient and begin the transcription at the correct site. But in contrast to the bacterial genes, the eukaryotic genes does not have specific terminator sequences. Also, in eukaryotes, after the transcription is completed, the transcript which is made first is not the final transcript. It's called as pre-mRNA. or premature messenger RNA, which has to be modified, processed to be converted to a mature mRNA, which can be translated into a polypeptide. So this is how eukaryotes differs from prokaryotes in transcription, and we will see this modifications and processing of pre-mRNA into mature mRNA in detail in the next few lectures. So let's in detail talk about the promoter sequence in eukaryotic genes. And the promoter sequence in eukaryotic genes have core promoter elements and promoter proximal elements. Let's talk about first the core promoter elements or the sequences in the promoters of the eukaryotic genes. What are core promoter elements? Core promoter elements are the genetic sequences or elements needed for the transcription machinery to start synthesis at the correct site. So how we have seen in the previous lectures in case of E. coli also, that once the RNA polymerase holoenzyme of E. coli orients itself at the promoter, the enzyme comes to know which is the template strand, in which direction the enzyme has to move, and where is the +1 nucleotide, the nucleotide which will be the first nucleotide to be copied into mRNA. So where to begin? The core promoter elements present in the promoter sequences of the eukaryotic genes are the elements needed for the transcription machinery to start the RNA synthesis at the correct site. Where are these core promoter elements? These core promoter elements are no more than 50 base pair upstream. So let's say that if I draw the promoter sequence here again, let's say this is a eukaryotic gene. And what I've drawn just now in green is the promoter sequence. And this is the RNA coding sequence from where the +1 begins. And where are the core promoters? So we have told in the previous lectures that the first nucleotide to be copied into the mRNA is +1, where the coding sequence begins. Before +1, numbers are assigned negative numbers. And this core promoter elements are no more than 50 base pairs upstream. So that means from +1 to -50 within this, the core promoter elements are present. The best characterized core promoter elements in eukaryotes are a short sequence called INR. INR is for initiator, and this initiator is present or span near the transcription start site. So it is present near +1, or it spans in the area near +1, which is the transcription start site. The other core promoter element which is best characterized in eukaryotes is the Tata box or Tata element, also called as Goldberg-Hognes box, named after its discoverers. So it has three names: Tata box or Tata element or Goldberg-Hognes box. And where is it present? This Tata box is at -30. 30 nucleotides upstream from +1. And this is called as Tata box because of this consensus sequence 5' T A T A. So Tata, that's why it's called as Tata box or Tata element. And then again, there are 3 adenine nucleotides. So this is 1, 2, 3, 4 Tata, 5, 6, 7, 3A. This is a 7 nucleotide consensus sequence called as Tata box. And this INR and Tata box do the same thing what -10 and -35 sequences, consensus sequences do in E. coli promoters. That is to help the RNA polymerase begin the transcription at the correct site. So these INR and Tata elements will specify where the transcription assembly, transcription machinery will assemble, and where the transcription will begin. So we had discussed only -10 and -35 consensus sequences in E. coli, whereas in eukaryotes, we have discussed the core promoter elements. But then we have to discuss something else also, which is equally important, and that is promoter proximal elements. Yes, INR is important. Yes, Tata box element is important. And these will tell RNA polymerase and the transcription machinery where to assemble and where to begin the transcription. That is, where is +1. But without other elements, transcription can occur at a very low level. So now let's talk about promoter proximal elements. When I say promoter proximal elements, they are a bit far away from the site where the transcription begins, that is +1. And as I said, if the core promoter elements are no more than 50 base pairs upstream, that is from +1 to -50, within that core promoter elements are there, INR and Goldberg-Hognes box. INR is near +1, Goldberg-Hognes box is at -30. In contrast, promoter proximal elements are a bit far off from +1, which means they are within -50 to -200 nucleotides from the start site of transcription. So the promoter proximal elements will be from -50 to -200, between this. There are two promoter proximal elements. One is cat box. The consensus sequence is CAAT. There's a cytosine, two adenines, and a thymine, and that's why it's called as cat box, named after its consensus sequence. And the cat box is located at -75. Cat box is located at -75. And there's a GC box. It's called as GC box because it is rich in guanine nucleotides. There are 5' to 3'. The consensus sequence is GGGCGG. So this GC box is located at -90. So the promoters in eukaryotes contain different combinations of core promoter elements and promoter proximal elements. And promoter proximal elements are important because promoter proximal elements are gonna tell when and how the gene will be expressed. And if mutations are introduced in the promoter proximal elements like cat box and GC box, if mutations are introduced in cat box or GC box, which are promoter proximal elements, then the transcription is reduced or decreased quite a lot from +1, which signifies that promoter proximal elements play a very important role in the transcription in eukaryotes. Let's talk more about initiation of transcription in eukaryotes, that is transcription of the protein coding genes that gives rise to messenger RNA. Accurate initiation of transcription of a protein coding gene in eukaryotes involves RNA polymerase II. We have seen in this lecture only that there are 3 types of RNA polymerases in eukaryotes, and out of the 3, it's RNA polymerase II that synthesizes mRNAs or carries out the transcription of the protein coding genes. But accurate initiation of transcription of a protein coding gene in eukaryotes involves the assembly of RNA polymerase II and other proteins called as GTFs. GTFs stand for general transcription factors. General transcription factors, as the name suggests, general and transcription, they are involved in transcription, and factors, they are proteins. So RNA polymerase II alone cannot do the transcription of the protein coding genes in eukaryotes. These GTFs play a highly important role, and they are important in a way, they are mandatory. So GTFs also assemble on the core promoter. We have just now seen the core promoter elements. In contrast to the bacterial RNA polymerase enzymes, none of the eukaryotic RNA polymerases can bind directly to DNA. So I will draw the promoter sequence again. Let's say this is a gene. This is a promoter sequence. And this is the RNA coding sequence. let's say if this is a bacterial gene and bacterial coding sequence and bacterial promoter, the bacterial or E. coli RNA polymerase holoenzyme can come and directly bind to the promoter, where it will contact the -35 and -10 sequence directly and will come to know where to begin the transcription, which we have seen in detail in previous lecture. So the RNA polymerase holoenzyme having alpha, alpha, beta, beta prime, omega, and sigma factor doesn't require help from any other protein. But let's say that for a second, this is a eukaryotic gene with a eukaryotic promoter and the coding sequence, and this is the eukaryotic RNA polymerase. This cannot bind on its own to the DNA. So the RNA polymerase of eukaryotes say, yes, I can do the transcription, but I cannot go and sit on the DNA, sit on the promoter on my own. I need help. So this is a eukaryotic RNA polymerase with a pol 1, pol 2, pol 3. It cannot bind to the promoter on its own. In contrast to the bacterial RNA polymerase enzymes, none of these eukaryotic RNA polymerases can bind directly to DNA. So the first step is that a transcription factor or a general transcription factor, GTF, comes and binds at the promoter. And I'll erase this and redraw this, because this general transcription factor or GTF sits at the promoter but near the +1, that is where INR is there and Goldberg-Hognes box is there at -30. So it assembles at the core promoter elements, and then this GTF will recruit some more GTFs, and then they will recruit the RNA polymerase enzyme to come and sit. So RNA polymerase enzyme needs help and it cannot bind to the promoter on its own. GTFs first bind, more GTFs bind, and then the RNA polymerase is recruited to form a complex. Even more GTFs bind after the RNA polymerase binds, and once the complex is complete, the transcription can begin. So this shows that how complex initiation of transcription is in eukaryotes. The GTFs or general transcription factors are numbered for the RNA polymerase with which they work and are lettered to reflect their order of discovery. For example, TF2D. TF2D is a transcription factor or a general transcription factor. TF stands for transcription factor. This Roman 2 stands for which RNA polymerase it works with. If it's 1, it works with RNA polymerase 1. If it's 3, it works with RNA polymerase 3. Since it's 2, TFT2D works with RNA polymerase 2 and recruits RNA polymerase 2 at the core promoter element for transcription to begin at the correct site. And the last letter tells us the order of discovery. If this is A, TF2A is the first transcription factor to be discovered. B, it's the second one. And since it's D, TFT2D is the fourth general transcription factor to be discovered, and it recruits and works with RNA polymerase 2. So in this lecture, we have begun with transcription in eukaryotes. We have seen how in eukaryotes the transcription is complicated as compared to bacteria, the 3 types of RNA polymerases, and they carry out the transcription of which genes, they synthesize which RNA, and then the promoter sequence. And the promoter sequence in eukaryotic genes consists of core promoter elements and promoter proximal elements. We have seen the significance of core promoter elements and promoter proximal elements, and we have seen or studied core promoter elements such as INR and Goldberg-Hognes box, and promoter proximal elements such as CAT box and GC box, and how promoter proximal elements are also important. And if they're eliminated, the transcription would be reduced drastically or will suffer a lot. And then we have seen how the initiation of transcription differs from bacteria in a way that the eukaryotic RNA polymerases cannot bind to the promoter on their own, and require the help from transcription factors.